This is a production of Cornell University.
So thank you for the nice introduction and the invitation. So actually, I'm really excited to present my work here at Cornell University. So when I was a graduate student, Cornell was my dream school. So I could not imagine I can, present a talk at Cornell University. So I'm really excited. So today I'm going to talk about some of my recent research focusing on transposon silencing, and the other area in my lab is working on meiotic recombination. So I'm trying to connect these two together. So that's what my title here is, transposon silencing and how transposon affect meiotic recombination in maize. So I took this figure from Citrix lab. This review actually tells you 10 things you should know transposable elements. But what I want to summarize that from the 10 things, the major conclusion is that transposons are really important. So they are not only junk DNA that you just try to filter and then ignore them. They are really important. They can create mutations. They also can regulate gene expression. So they are involved in many diseases. So transposons are really important. And as you know that most transposons, the T are the major components of many eukaryotic species, like the human genome. Nearly half of your DNA are transposable elements. And then the maize genome, over 85% of the DNA are transposable elements. So, although transposons are the most abundant component in many eukaryotic species, most of the transposons are silenced. So that's silenced by epigenetic pathways, including like demethylation, histone modification, and non-coding RNAs, for example, small interference RNAs. So if you pick up transposon in a genome, most of the time you will find it is silenced. So it is methylated or either methylated or histone modified. So they can't move anymore. So that's what we find right now. So in plants, if you work on plants, you may know this RNA-directed demethylation pathway. So, you can see plants has two specific polymerases called POR4 and POR5 here. So you can see POR4 and POR5 here. So this POR4 can transcribe DNA into a single-strand RNA. So then this single-strand RNA can be copied into double-stranded RNA by RDR2 here, RNA-dependent RNA polymerase 2. So then this double-stranded RNA can be cut into small RNAs, and then loaded back to the original position or the regions with similarities to trigger de novo methylation. So this is RDDM pathway. So this RDDM pathway can trigger all of these three different types of methylation in plants: CG, CHG, and CHH. We know that in mammals, you may have the major one is CG methylation, right? So in plants, we have pretty abundant CG, CHG, and CHH methylation. So these three different types of methylations, they are maintained by different methyltransferases. So for example, CG is maintained by MET1, and the CHG is maintained by CMP3, is a chromatin methyltransferase. And the CHH in heterochromatic region is maintained by CMP2, but all of these three can be triggered by RDDM. So if you take a look at the maize genome, you can see they are near 65% of the cytosines are CHH cytosines. So which means CHH cytosines are really abundant. However, if you check the methylation level, so you can see CG methylation level can up to 84%. CHG methylation level can up to 72%. But the CHG methylation globally only 2.4%. So it's very low methylation level globally. But our lab is pretty interested in this small level of methylation. So later I will show you why we are interested. So although CHH methylation is genome-widely very low, it can be enriched into a high level near genes. So you can see here is a transcription start site, here is a transcription termination site. You may notice that there are two peaks here. that we call CHH islands. You can see that the methylation level of the CHH can up to even 10% or 12%. And people also find that the major, inducing this CHH island are transposons. So you can see you have a transposons here. You check the ages of the transposon, you will find that actually these ages of transposon are methylated, which form these islands. So you can see here the methylation level even up to 20%. And that's why people propose that this island actually mark the transition from heterochromatin to genicly-active euchromatin. So they propose it's a transition to separate heterochromatin here and the euchromatin here to prevent there are a lot of still arguments. One argument is that this island is trying to prevent the heterochromatin effects. The other argument is this island is trying to protect the gene expression to flanking transposons. So we are pretty interested in understanding, how this CHH methylation is initiated, how it's maintained to next generations. So that's why we have been using a mutant called MOP1 in maize. So MOP1 is in maize, which is the ortholog gene of RDR2 in Arabidopsis. So actually, in this MOP1 mutant, you find 24 nuclei small RNA are largely reduced. So you can see this is a wild type, and then this is a mutant. And then you also can see at the CHH island region, methylation is largely reduced. So you can see, right? So in addition to using this mutant, we also use hybrid to test how silencing is initiated and maintained. So, for example, you have two different maize backgrounds or different parents. So they are methylation is differentially methylated. So you can see at this region, parent 1 is highly methylated and the parent 2 is unmethylated. So they are separate, right? But when you hybridize them together, these two sets of chromosomes, they are in the same nucleus. So how their methylation pattern look like? So we propose 3 different models. For example, the first one, the methylation does not change. So you can see allele 1 inherits methylation from parent 1. Allele 2 inherits methylation from no methylation from parent 2. So then we call this no change. But there are many situations these two chromosomes can interact with each other. So you can see this allele 2 gains methylation, although parent 2 does not have methylation at the same locus. And then this is we call transchromosome methylation, TCM. And the other situation is, parent 1 has methylation here, but in the allele 1, methylation is lost. So we call this transchromosome demethylation. So when we have these different patterns of methylation or demethylation, we also want to check whether this kind of gained methylation or remove methylation can be maintained to next generations. So that's why we also examine the F2 generation. So in order to do this, I hybridized this mutant heterozygous in the MOR17 background with the heterozygous MOP1 in the MOR17 background. So then you can create a hybrid. Also this is a mutant hybrid, and then this is a wild type hybrid. So then you can compare combine the mutant effects and the hybrid. So then later on, we performed the whole genome bisulfite sequencing of the two parents and their F1 hybrids. So we did whole genome bisulfite sequencing. So here just shows you one chromosome 1, maize chromosome 1. So this is a physical distance, and then this is a demethylation level. So you can see that for CG and CHG methylation, they are pretty stable, no any large difference genome widely. But if you check the CHH methylation, so you can see these two are the parents. And then this one is a wild type hybrid. So you can see actually hybridization increased a lot of genome-wide CHH methylation. And even, so this is a MOP1 hybrid. So we can see this MOP1 hybrid, the CHH methylation level is still higher than the two parents. So this data suggests that, although this mutant, we know it can remove methylation. So actually the effects is not as strong as the hybridization. So hybridization even triggers more methylation than the mutant, the removal methylation. So, this is a genome-wide analysis. So then later on, we focus on differentially methylated region we call DMRs here. We want to identify the parental DMRs. So you can see this, this parent has higher methylation. We call this is a B73 hyper DMR, which means B73 has higher methylation. So then we want to see, how this parental DMR, located or how their pattern looks like. So we identified over 7,000 CG DMRs, 9,000 CHH DMRs, CHH even up to 13,000. So these are between the two parents. And if you check these two different maize background lines, you will find B73 has more hyper CG and CHH DMRs, and MO17 has more hyper CHH DMRs. This also indicates these two maize lines are pretty different. And then this is also consistent with the methylation level. You can see B73 has higher CG and CHG methylation, and MO17 has higher CHH methylation. And we also check the location of these parental DMRs. We also find that these CHH DMRs are different with CG and CHH DMRs. So you can see these CHH DMRs are largely located in the 2 kb upstream and downstream region. So which means these CHH DMRs are near genes, Over 43% of the parental CHH DMRs are in the 2 kb upstream and 2 kb downstream of genes. So then we want to check, we identify these parental DMRs. We want to see how their methylation level look like in the hybrid when we combine these two sets of chromosomes together. So we find that the majority parental DMRs, they don't change their methylome. Even you brought up these two sets of chromosomes together. Only 25% of the parental DMRs change their methylome in F1. So out of this 25%, I think about 19% are TCM, which means they gain methylation after hybridization. There are also 7% are TCDM, which means methylation is removed in those regions. And next, we want to see, we observe this increased methylation at some region and the decreased methylation in some other region. We want to see which allele actually changed methylation. So that's why we compared high parent with low parent, high allele with low allele. So this here high parent means the parent has higher methylation. So you can see here, you can see average, this is a parent, this is F1 hybrid, and then this is MOP1. Hybridization at this TCM actually induced a lot of methylation. So when you separate them into different alleles, we find that for CG and CHG, the high allele do not show significant difference with high parent. Only the low allele, low parent allele, it gains methylation for CG and CHG methylation. But if you check CHH, it's different. This CHH actually both the high allele and the low allele, both of them gain methylation. Although the high allele is very high in the parent, it still gains more methylation after hybridization. This also shows the CHH methylation is different with CG and CHG methylation at the TCM DMRs. For TCDM, methylation is removed. This is pretty consistent for these three different methylation. You can see all of them high allele reduced. But for TCM gain methylation, CHH, both allele gain the methylation in our data. So we also check this MOP1 mutant effects. We find that this mutant actually mainly functions on CHH methylation. So if you can see in the mutant, the change the methylation, you can see the TCM out of this 1,040, probably 1,043 TCM DMRs, more than 90% the methylation were removed. Even you gain the methylation in your F1, the methylation is removed by MOP1 mutant. For CG and CHG, we identified only a few of them changed the methylation after in the mutant background. So if you check the methylation level at this affected TCM, you find, CG, CHG, they are reduced the methylation in the mutant, but the CHH methylation even removed to 0. You can see even lower than low parent allele. So this data, suggests that, or at least indicates MOP1 mutation largely works on CHH methylation. So as I showed you the RDDM pathway, we know that the small RNA are the trigger of DNA methylation, right? So then we wonder, in F1, it gain the methylation. So whether small RNA are produced or not. So for example, we proposed 2 different models. So you can see parent 1, parent 2. Parent 1 has methylation, so it has small RNAs produced, and the parent 2 does not have small RNA. And then when we combine them together, probably allele 2, produce small RNA. Although parent 1, parent 2 does not produce small RNA, but after hybridization, it may produce small RNA. And the other hypothesis is, the small RNA are still produced from allele 1 and then trigger methylation in trans. Small RNA don't have to be produced again, but the small RNA from parent 1 can trigger methylation of allele 2 when you hybridize them together. To test these 2 different hypotheses, so we performed the whole genome small RNA sequencing from the same tissue, and we find that, consistent with previous data, MOP1 actually largely removed 24 nucleotide small RNAs. And if you check the parents with F1 hybrids, we don't observe very significant change regarding small RNAs. Small RNAs are pretty similar. This is a genome-wide analysis. Then later on, we compare the high parent with low parent. High parent means the parent with higher methylation. So typically, if you have higher methylation, you have more small RNAs. We find high parent has significantly more small RNAs than low parent. And if you compare the MPV value, MPV is the average level of small RNA between of high parent and low parent. If you compare the MPV value with F1 hybrid, if you check small RNA, we don't observe a significant increase in small RNA. Of course, MOP1 removes a lot of small RNAs. And we also check the ratios, between the high parent and low parent, and between the high allele and the low allele. We also did not observe significant difference. So all of our data suggests that small RNA are still likely produced from the original allele, that generates small RNA. So that, and then trigger methylation in trans. So we think our data supports this hypothesis, small RNA are still produced from the original allele and then trigger methylation in trans. So next we want to see, we have this gain of methylation. We want to see whether this methylation affects gene expression. So as I explained to you, this CHH methylation, many people find it promoter methylation can suppress gene expression. But in some other research, they also find that CHH methylation is associated with higher gene expression, probably because of the boundary. We don't know which one is true. So that's why we proposed the 2 different models. The first one is if CHH methylation is associated with suppressed gene expression, and then you can see you have this methylation in the promoter region, and then this parent has produced less transcripts compared to the other allele. Another parent has lower methylation, right? So after hybridization, this allele gains methylation. So if CHH methylation suppress gene expression, we should find that this ratio increased. It's hard for us to directly compare the expression level, mainly because, in the F1 hybrids, the 2 sets of chromosomes are together. We only can use SNP containing reads to distinguish the expression values. So that's why we use a ratio here. So the other is if CHH methylation promotes gene expression, we may find that this ratio decreased. So then we, based on these 2 different models, we try to find any gene matching the first model or any gene matching the second model. So we only find 16 genes show the first model, and then 31 gene, shows the second model, which means that this 16 gene actually CHH methylation suppress their gene expression. And then here, this 31 gene, CHH methylation may promote their gene expression after gaining methylation. So then we were pretty interested in comparing these two sets of genes, why they, this 16 gene was suppressed by CHH methylation, but these 31 genes were promoted by gene methylation. We compared a lot of parameters, and we find actually this 31 gene, the second group, actually the gene length is longer, significantly longer for this 31 gene. And then gene expression also higher. And also gene body methylation, this 31 gene is also higher. And we also compared several histone modification marks. We find actually this 16 gene are suppressed by CHH methylation. They have very high H3K27 trimethylation. So as you know that this mark is a suppressive mark, right? So for those genes are suppressed by CHH methylation, they also have higher histone marks. So we are still not very clear how these two sets of genes are different, but at least we show they have many features different, including gene length, gene expression, gene body methylation, and H3K27 trimethylation. So next, we also wanted to ask, whether this triggered methylation can be maintained to next generations. So that's why we backcrossed our F1 to one of the parent, and then we sequenced the whole genome methylation level, and then check at those regions when methylation gained in F1, how the methylation pattern looks like in backcross BC1 individuals. So we also have CG-CHH data. So here just shows CHH. So our overall conclusion is that CG and CHG methylation are kind of overall good, better maintained to next generations. But the CHH methylation, even you gain the methylation after hybridization, in the next generation, the methylation also reduced. So the maintenance is not very stable. So this is the overall data. Different genotypes are very complicated, but overall we found the CHH methylation is not better maintained to next generations. So this is my part one. I want to summarize a little bit. We find hybridization increased the CHH methylation globally, and we also find at the TCM DMRs, gain methylation, we find CG-CHG. The gain methylation is mainly caused by the increased methylation of the low parent allele. But for CHH methylation, both high and low parent alleles are increased in F1. We also find MOP1, works on mainly CHH methylation. And we also find small RNAs are still produced from the allele that originally produced small RNA and the trigger methylation in trans. And we also find CHH methylation, is associated with both suppressed and enhanced gene expression. We find the newly triggered methylation can be maintained to next generation, but it varies in different sequencing cytosine contexts. So I become interested in meiotic recombination. Actually, starting working with Damon, he works on transposable elements. So when I joined his lab, I read some papers, and most of the papers find the recombination is negatively correlated with transposon density, right? So if you have a transposon there, normally this is not a hot spot. So typically you have a negative correlation between recombination and transposons. So that's people also find in autopsies, recombination rate is positively correlated with gene density, positively correlated with H3K4 trimethylation, and positively correlated with low nucleosome density, but negatively correlated with demethylation. So that's why, if we remove methylation, I also want to ask whether we can increase methylation or not. So the other thing we know during transposition, transposons create double-strand breaks, right? So you can see this just the example of mutated transposon. When it jumps out, it creates a double-strand break. And we know that the first step for meiotic recombination is produce double-strand breaks. So that's why at that time I want to ask whether these bricks generated by the transposition of transposons can be used to create more crossovers or not. So can these bricks be used here in the meiosis pathway to create more crossovers? So that's the main question when I was a postdoc. I want to check how active and silent transposon affected meiotic recombination. So the first one is either due to double-strand break when they are active. So for example, if you have an active transposon here, then you have a silent transposon. When this transposon jumps out, it creates double-strand breaks. Can these breaks be used to form more crossovers or not? And then, or due to demethylation, they bring to genes when they are epigenetic silenced. As I showed you, for those CHH islands, they are mainly caused by transposon insertions, right? If there's a transposon there, that transposon needed to be silenced. So then probably when you have a hot spot, so you can see the gene is a hot spot. And then when there's a transposon jump near to the gene, and then this transposon get methylated. So whether this cold spots become, cold spots or not, or you can change another way. If you have a cold spot, if you remove this transposon or remove the methylation, can you increase more crossovers or not? So these are the central questions that when I was a postdoc, I want to understand how active and silent transposon affect meiotic recombination. So to test the first question, to test how active transposon affect meiotic recombination, we have an active mutated line in maize. So we also have another silenced population. So in Damon's lab, he has a mucala. When this mu transposon is active, we can easily silence it by adding this by introducing this mucala into that line. So then we have an active transposon population, and then we have a silenced population. And then for genetically, they are identical because the insertion is in the active population, and then also this insertion is in the silenced population. So then we can examine the recombination around this active mu insertion and compare the recombination around this silenced transposon. So now we only have one locus data. So we clone the one locus. This mu, we know where it is. So then we design a marker and then examine the recombination around this active mu and then this silenced mu. We find that this active mu, actually the centimorgan can up to 18 centimorgan. The silenced mu only 12 centimorgan. So at least this locus is, as we expected to see, active transposon. When you have an active transposon there, you have a higher recombination rate. My lab also, trying to clone all the mu insertions and compare all the locations, how recombination differed between active mu and silenced mu. The other question is, how do silenced transposon affect meiotic recombination? So you can do two different ways, right? You can try to induce an insertion and then to see that hot spots become cold spots or not. Or in another way, you can remove that transposon or remove the silencing of that transposon to see whether this gene or we can create more hot spots or not. So I use the second one, because I have this MOP1 mutation. I know it can remove methylation near genes. So that's why when I was a postdoc, I hypothesized that in this mutant, we may observe increased methylation because this mutant can remove, it can increase recombination in the mutant because this mutant can remove demethylation. Demethylation is kind of suppressing recombination, right? So that's the central hypothesis at that time. But actually the data was even more interesting than we expected to see. We expect to see we can observe more crossovers, but we find actually we only observe more recombination in the chromosome arms. So you can see these are the 10 chromosomes, chromosome 10, 1 to 10, and then these are chromosome arms, and then these blue bars are pericentromeric regions. And then above 0 means increased methylation in the mutant, and below 0 means decreased methylation in the mutant. So you can see out of the 10 chromosomes, 9 chromosomes increased methylation in the chromosome arms in the mutant. And out of the 10 chromosomes, all of them were decreased, recombination were decreased. So if you do the statistical analysis, they are significantly different. So next, those are just a few markers. So then later on, we developed more markers across 6 maize chromosomes. So you can see above 0 means increased methylation. You can see almost in the mutant, the chromosome arms recombination are increased. So you can see here, and all in the pericentromeric region, recombination were decreased. This is pretty consistent as our data. And also, we did a cytogenetic analysis using antibody of HEI10. This gene tracks the crossover precursor. So this just shows one myocyte. So this is a wild type, and this is a mutant. If you see a red dot there, that means one crossover precursor, one HEI10 focus. So we didn't see very big difference. So overall here probably 19 and here probably 20. We didn't see very big difference. And also later on we traced the one chromosome, chromosome 6. We divided the chromosome 6 into 10 equal bins, and then calculate HEI10 foci across many different myocytes. So you can see in bin 10, in the mutant, the HEI10 foci is largely increased, right? So overall, this data suggests that the crossovers are overall unchanged, but occurred more frequently in the distal end of the chromosome. If the crossover events are just redistributed in the mutant, it increased in the arms and then decreased in the pericentromeric regions in the mutant. So this is pretty similar as a research similar as a MET1 mutant in Abdopsis. So later on, we also want to understand, why this occurred. So that's why we sequenced the whole genome demethylation of the mutant and the wild type. So normally you can see CHH methylation, you can see is higher in the mutant, in the wild type, and compared to the mutant, right? CG, CHG do not show significant difference between the mutant and wild type. And then if you check the island region near genes, you can see the mutant largely remove this island. And then if you check the transposon, you also can notice the ages of transposon methylation also reduced. So we think, probably this mopi mutation dramatically reduced methylation near genes that result in the redistribution of crossovers. So actually, later on, my lab also did a high throughput sequencing. We try to create a high resolution crossover map. So that's why we sequence the 90 backcross individuals, whole genome sequence, and that derived from the mutant, and the over 100 derived from wild type. We try to use all the SNPs to limit the crossover at a very small region. So we just got the data. We identified over 1,100 crossovers derived from MOP1, and then 1,400 derived from the wild type. And then if you plot out this crossover, you can see many chromosomes in the distal end. For example, chromosome 6, there are a lot of more crossovers in the distal end in the mutant. But we are still not finished analyzing the data. We try to identify all the crossovers, probably focus on the crossovers within 2 kb, and then to see which region indeed crossover are changed. So for MOP1, all the data I showed you right now are from MOP1 being used as female. So we also did a comparison between male and female. So if you if you see here, above 1 means methylation increase, recombination increased in the mutant. So you can see overall, if you compare male and female, they don't have significant difference. However, if you separate, the chromosomes into chromosome arm and pericentromeric region and compare the wild type and the mutant, you will find actually in the mutant, recombination got a larger increase in the female and a larger decrease in the pericentromeric region. So this data is pretty preliminary. We also sequenced almost another 100 or 200 backcross individuals derived from the male. We want to compare female and male, how their recombination differed. And we also want to examine how demethylation, affects female and male differently. So actually from this data, it inspired me, let me ask how, methylation differs between sexes in the maize population. So that's why I developed another project trying to compare recombination between sexes in the maize population. So you can see the diversity of maize is amazing. You can see there are so large diversities. So then later on, we use the 25 name finder lines because they are pretty diverged. So they can explain 85% of the maize population genetic diversity. So that's why we use these 25 name lines. And then we want to compare female and male meiosis. So that's why we, cross these 25 name lines with B73, and then we created this F1 hybrids. So then we use this F1 hybrids as female, and meanwhile use this F1 hybrid as male to produce backcross populations. So then these two different backcross populations can use it to estimate recombination occurred in F1. So until now, we did a whole genome genotyping by sequencing. We found the data is really interesting. Until now, we screened 9 lines. And out of these 9 lines, we found 3 of them have a higher male meiosis. So you can see B97, Ohio 43, and IL14H. This is a sweet corn, and this is a temperate maize. So these 3 lines have a higher male recombination. And the other 5 lines do not show significant difference. And the last line called CML103, it has higher female meiosis. So this data is pretty interesting to us. We thought probably some lines are different, some lines are not different, but overall, out of these 9, we identified 3 have a higher male meiosis. So if you check their recombination rates or genetic distance, you can see it varies. Although B97 has higher male recombination, also it varies in different chromosomes. You can see chromosome 7, chromosome 8, chromosome 9, they are very small difference, very little difference, but other chromosomes are very big difference. And also you can notice that in the distal end, recombination is kind of very higher, or recombination rate is higher in the male. So you can see chromosome 2, chromosome 4, 6. They are all, overall recombination is higher in B97 hybrid, and especially in the distal end, in the telomere region. Many chromosomes, they have very high recombination in male. So if you check CML103, this one has higher female meiosis recombination rate. Typically, if you can see here, when it has higher female, it not only limited to the distal end, it across the entire chromosome. You can see even not near the distal end. So this data also shows us the difference, right? Normally, if you have a higher male recombination rate, either they are limited to the telomere region or distal ends. But when this line has a higher female recombination, it comes off across all over the chromosome, not only limited to several regions. So these are really not preliminary, but we still haven't finished analyzing the data. So then next we compare the female, all the recombination across different lines, across female and across male. So the data is very interesting to us. We found that, female recombination is kind of very stable. So you can see this female, we don't observe very large difference, even across different lines. So they are pretty stable, but it varies substantially during male meiosis. So you can see this male, different lines, B97 is very high, and the CML103 is very low, but the female is very stable. So overall, this data suggests that the meiosis recombination differs largely in different maize backgrounds. And also, this data tells us meiotic recombination is generally very stable during female meiosis, but varies largely during male meiosis. So we are not finished analyzing the data. We are also screening all the remaining probably over 10 lines. So after we screening, we try to identify which line has very large difference between male and female. And then the other one maybe has very high male, then we can send these two representative lines for whole genome resequencing. And then we can create a very high crossover, high resolution crossover maps to identify which region actually are different between female and male. So we try to build this high resolution crossover maps. We are also trying to isolate myocytes to do transcriptional analysis, small RNA analysis, demethylation analysis, and histone modification analysis. We try to integrate all of the data together to tell how sexes, the recombination differs between sexes in maize. So I want to summarize my second part. We found one active mutated insertion at the increased recombination. We also find the MOP1 actually decreased methylation in pericentromeric region, but the increased methylation in recombination in chromosome arms. We also find probably crossover redistribution is likely driven by rules of CHH methylation near genes. And we find the demethylation may be play different roles between male and female recombination. And we also find the meiotic recombination differs substantially between sexes in different genetic backgrounds. We also find meiotic recombination is generally stable during female meiosis, but varies substantially during male meiosis in different maize genetic backgrounds. So I would like to thank my postdoc advisor, Dr. Damon Leach. I initiated this project in his lab, and then he finally let me take this project to my own lab. So that's why we finally have a big project. So also I want to thank Professor Rachel Wine. She did all the cytogenetic work for our lab. We are a pure computation, not a pure. We do a lot of computational stuff. And I also want to thank Voltag. He reviewed my papers, reviewed my background proposals, which make my lab survive. Otherwise, I may not have people working for me. I also want to thank all the undergraduates and graduate students. They did all of the work. So my research is funded by NIH and NSF, and also the startup from my previous university, Miami University, and my current university, University of Florida. So with that, I would like to take any questions if you have. Yeah.
Wonderful talk. I had a question. Why do you think CHH methylation was increased in the hybrid, even with the MOP1 mutation?
Yeah, so we actually work on several different projects. We find actually CHH methylation is very dynamic. So for CG and CHH methylation, they are kind of very stable. Even you have stress, they may not change too much. But for CHH, they are very stable. So CHH is very dynamic. So that's why I hypothesize that CHH is kind of like a buffer. There are so many cytosines across the genome. Although they are very low level, but there are so many. So they can be served as a buffer, when stress come to the plants and then they change. But they can rapidly return to the normal level after the stress is released. That's just my hypothesis. But we have another project in my lab. we inoculate the plants with pathogen, we also observe the CHH methylation reduced, not increased in that experiment. But all CG and CHG, they are globally not changed too much. Of course, you can identify a lot of differential methylated region, but genome-wide, you don't see much difference after inoculation. But you observe genome-wide decrease of CHH methylation. So, and also another lab, they did a heat stress. They also find the CHH methylation changed rapidly. So what we hypothesized that this CHH is kind of like a buffer, try to buffer the changes of the chromosome to maintain the chromosome, for example, stability. That's just a hypothesis, but we observed that CHH methylation changed dynamically in response to different stresses, for example, or hybridization. Yeah. Mm-hmm. Thank you.
I was wondering, for the first part of your talk, did you break down the transmethylation patterns by TE family or subfamily and notice any like trends based on the specific like CHH sites that you were profiling?
So you mean those TCM DMRs in
Yeah.
the F1? Yeah, we checked the different types of transposons. When you see the majority are DNA type transposons at the TCM DMRs. So we have the data, they are in the supplemental. We submitted this paper. We checked all different kinds of transposons. The majority one is PIR transposons. And then next is helicron. And then next is LTR retrans transposon near the overlapping with the CHH.
Did like
TCM.
all the different families like behave the same though in terms of like changes in methylation pattern, or were there family specific differences?
We didn't do family level. Yeah, we did only PIR DNA transposon and LTR retrans transposon and helicron. I think we didn't do family specific level. Yeah, that's maybe a good point to see other if there is any specific transposons there.
Cool. Thanks for a great talk too.
Go to the back of the room and then we work to the front.
Yeah, thank you. Actually, I want to continue with the last question. How is when you increase the CHH methylation, what kind of specific T family are kind of triggered? Did you calculate all the T insertion transposition to know which kind of family are associated with CHH methylation?
So yeah, that's a good question. When we work on transposons, we base on the annotation. So I think that there are some things we can't do using our current data because we rely on the annotation. There may be some wrongly families, we don't know. So that's why we finally didn't do too much analysis on transposons. Initially we planned to do transposons, and finally we found that we have very low power to do every family or polymorphisms between MOR17 and B73, mainly because when we use the MOR17 genome, we use a SNP replace the MOR17 genome. Which means we are not focusing on the polymorphic transposons. If you want to use a de novo MOR17 genome, it's just a little bit computational challenging to identify which transposon is present, absence. So, and then we also want to tell different alleles. So then finally we decided to use a SNP replace the genome. We lost all the transposon polymorphism data. That's the weakness. I didn't want to point out because I don't want the reviewers to ask this question. But we know, we lost a lot of data regarding transposons because we use a SNP replace the genome. So that's why I finally decided to switch my focus from transposons to demethylation. My initial thought was trying to, compare this has a transposon insertion, this one does not have, how the methylation changed in F1, but finally we didn't get there. Probably in the future we can do that. Just the transposon, they are repeats. If you want to if you want to, and finally, actually, centrally, we work on perimutation. Many of you know that, right? We compare the same locus. Both of them are in the parents and in the F1 hybrids. We lost all the transposon polymorphism data using our computational approach. Yeah. So that's why I can't do a lot regarding which type of which T family, because all the annotations are based on B73. Yeah. Mm-hmm.
And thank you. And also one more small question for my own interest is that I also curious about how you can correlation the transposon with recombination events. So how you screen for this recombination event? Is you are using like molecular marker like SNPs to tracking down the location and frequent?
So yeah, we use black cross populations. You know, I indeed I have a slide. So see, we have, you can see you have polymorphic markers between these two backgrounds, and then in the F1 hybrid, when there's a recombination occur between here, so you have 4 different gametes, and then these 2 are the parental gametes, and then these 2 are the recombinant gametes. If you back cross, you will identify this kind of recombinants using the markers here. So that's why my lab we use back cross population to examine recombination already occurred in F1. Okay. Yeah, unlike Voltax lab, they can directly work on meiosis, then, you know, but we are not good at that. So that's why we use backcross populations to estimate the recombination already occurred here. Mm-hmm. Thank you.
Really cool. So thinking about the stuff you talked about at the end with the male-female differences in recombination, are there any differences in the inheritance of the small RNAs that are maybe responsible for targeting DNA methylation, and would that perhaps explain what's going on with recombination rates in developing pollen or ovules?
That's possible. So that's why we are going to do, you know, we are trying to sequence a lot of small RNAs or DNA methylation. But there's a problem here. I know probably Voltax knows that, you know, isolating female meiosis is really challenging. So we can't directly get the small RNA data or DNA methylation data directly from female meiosis. So we only have this data for male meiosis. So we don't have compare. But I propose that, you know, I think you can see here, for female and male, we observe difference. I think probably chromatin structure is really important between female and male because their DNA is same, right? So I think their chromatin structure in the female and chromatin structure in the male is very different. But we are going to probably for my next 30 years, I want to find out whether it is true or not. Yeah.
You already answered the question what I want to ask, but I have a second one. What do you think? Why the B97 have the increased number of the crossover? Do you have any ideas how it happened and why it happened?
We don't know. We don't have any idea. So you can see they are pretty mixed. I feel I previously saw probably some tropical maize, they are very large different. And the temperate maize, they are very large different. But it looks like you can see this is tropical maize, and then this one is also tropical maize. And then that B97, I think is temperate maize. So we don't know why they show difference.
But you also have B73 and not those.
Those are hybrids, yeah.
That might.
But I think this data is more interesting. We find the female is pretty stable. You can see we already screened 9 lines. Female, no big difference, no significant difference. Only male changed. Yeah.
Because what we found was that the reduce of the CG and CAG methylation increased the crossover frequency. That might be something what you can look. That means look on the methylation status of all of these inbreds.
Yeah, that's we plan to isolate them male meiosis, and then send for demethylation sequencing. And then we can compare B97 with all the other lines. Yeah, that's we are planning to do. We are still screening the remaining lines, but we already screened 9. We found the female is stable. I think this is probably the general pattern, and the male is very diverged. Yes.
Hi, great talk. Is this on? Yeah, okay. I was actually gonna ask about this, so it's perfect that you're on this. I noticed that I can barely read it from back here, but K11 or KL11, that kind of behaves like a male. Do you have any ideas of how you might be able to use?
You mean KI11?
On the other side, on the
KI11.
female side, yeah, it kind of behaves like a male where it has that longer distribution, and then there's other ones on the male side that have a very short distribution.
Yeah.
Do you think that you can kind of use these as a way to figure out why there's such a tight distribution in the females and such a wide distribution in the males? Like, what's driving it, or are these like standing out as a special thing? And the same thing I think was true for, do you have a hypothesis for why the meiotic recombination rates from the female parental side are so wide over the chromosome, but in the male side are so tight to the end?
That's a good observation. I even didn't notice that. So, our data is pretty. We don't have enough data, so we are still working on that. So as I mentioned, after we screen all the 25 lines, we will send for high throughput sequencing. So then we can identify crossover at high resolution. So then we can compare all the DNA sequences or DNA methylation data. So when we have all of these different types of data, I think we may have a better idea, why female is so stable and the male varies, and what kind of parameters is the most important. Yeah, we don't have too much idea right now. Yeah. Thank you for your question. Thank you for pointing this out.
This has been a production of Cornell University, on the web at cornell.edu.